Okay, welcome everybody. I believe we left off right after the video. So close to wrapping this chapter off. I apologize. I realized there weren't any other lectures posted until just moments ago. So if you were hoping to see stuff before, and I'm pretty sure you weren't, but if you were, it just got posted a couple of minutes before class. So I do have all of the other PowerPoints up on there now. So that should be smooth sailing. We will move into the next chapter here in just a minute. We have just a few chapters to cover before the next test, which I think might be a little better, might be a little more focused with 3 instead of 5. Hopefully that will be better. I will tell you exam stuff. I have started my process of reviewing. There are a handful of pages with everybody that I need to go through. I'm almost through the first page, and I think there are 3 total. So I don't know how many people on each page, but I have looked through most of it. So far, so good. Any questions or concerns? And if you're tuning in from home, unmute yourself. Okay, guys. So we talked about cell cycle. We've shown some ideas with it. We're going to talk about bacteria as we wrap this up in a second. But we've been thinking about eukaryotes and how cells, normal body cells, go through the process of replication to generate identical cells. I said that over and over. They said it over and over in the video, right? And that, I mentioned different resources, Khan Academy and whatever. Amoeba Sisters has a plethora of biology videos on YouTube if you are interested in that stuff. And I'm pretty stinking sure we will slap a few more of them up here. But they definitely have more stuff out there. So if you're interested and you thought they were pretty good at describing it, which I thought they were, I recommend you check some of that out. So what we talked about last time was normal stuff, how normal cells go about the process of replicating and generating identical cells, right? That is what we would like to have happen all of the time, but that's not 100% of what happens. Sometimes cellular replication goes haywire, goes kind of nuts when we think about that. I should have manipulated this video, not that you want to be seeing me in this stuff, but it's all sorts of wonky now. I should just stop moving. Maybe that would be better. Okay, so with that, when we think about cancer, this is when cell division basically goes unchecked or it's uncontrolled. There are all sorts of potentials and situations that set the stage kind of for the perfect storm for these issues to really result. Oftentimes we think about mutations, changes in the DNA that impacts cell division. And we talked about before the video, there's a handful of mechanisms in the cell cycle that are checkpoints that help the cell to ensure that stuff has happened properly, or if it hasn't happened properly, we potentially manipulate stuff and allow it, longer time to deal with it, or those cells get targeted for removal. If we get past that first restriction point, if there's other issues, typically that cell's gonna get pulled out of the circulation, taken out of commission, right? But with cancer, that stuff doesn't happen, right? That's where the issues ultimately show up. We start seeing mutations in the parts of the cell cycle that are these checkpoints. And when we no longer have the crossing guard standing there allowing things to occur or stopping them, it goes unchecked, out of control, all right? So we'll go through and talk about some of that. We only have a few slides to evaluate with this in the first place, so it's really not a whole lot. We'll talk about specific genes, and when they get altered, again, mutating, it's like a switch, a trigger that turns on these mechanisms that are technically being held in check, all right? So a proto-oncogene is a gene that ultimately is functioning properly, and then upon mutation, you drop the proto off and it becomes an oncogene. Oncogenic genes turn on massive cell growth, if you will. It stimulates tumor production, along with the suppressors. There are other genes that help to inhibit this stuff, and when they get altered or mutated, what should be stopping no longer does. It doesn't suppress anymore, right? So we will see a little of that right here. So again, normal situations, everything functions properly, right? And it's when there's some sort of alteration, some sort of mutation, when we end up not functioning as we should. So we've got two examples to try to show this. The normal situation, everything functioning as it should, and then through mutations, no longer, right? So this protein up here, normal P53 protein, it's going to basically help to monitor and do surveillance to ensure that stuff in the cell cycle is happening properly, right? It's monitoring DNA. And if there are issues, if it detects some types of mutations, not every mutation, not every alteration in the DNA gets recognized by P53. It's specific things where maybe we don't base pair properly. We end up causing kind of like little speed bumps in the genetic material to recognize that type of stuff, right? Other issues, but a simple base pair mismatch, that goes undetected, right? So if the damage is detected, this protein, P53 protein, triggers repair mechanisms. It recognizes there's problems and let's fix this stuff, right? If we can fix it, it's all fine and good, right? If that's not the case, and you can't repair this damage, then it signals another mechanism. Apoptosis is programmed cell death, cell suicide, right? And these cells that are messed up, you know, technically they take one for the team. They destroy themselves instead of proliferating and causing all sorts of other issues. So the P53 protein, it recognizes the damage in the DNA, it recognizes specific abnormalities in the cell cycle, along with hypoxia. So low levels of oxygen, and that can cause other issues as we think about metabolic processes, setting the stage for problems inside of the cell. So it can slow the cell cycle, halting it, giving it time to potentially deal with some of these issues. It can correct some of the, again, mutations that have happened, specific ones. And it can also, once we have stopped the cell cycle, if we've done stuff and fixed it, it can reinitiate it and say, okay, the crossing guard is dropped. We can move through and proceed in to the rest of this. If none of this happens, we can't correct stuff. That's when the apoptosis happens, and that cell ultimately gets destroyed. That's the normal situation. That's what we want to have happening inside of our cells as a control mechanism to ensure that these things help, if you will. If this protein is mutated, the P53 gets mutated, now the mechanism to monitor and do this surveillance is basically thrown out the window, all right? So it doesn't repair the damaged DNA. It doesn't signal apoptosis. It's like, nope, we're not removing you. We're going to let you persist, right? And that sets the stage for cancerous situations. It sets the stage for tumor growth, unchecked cell proliferation, lending to the possibility of cancer, right? So here it's showing that it's altered, it's messed up in some way. It no longer is able to do its job of detecting, and the cell cycle doesn't get stopped if there are issues, it just continues, and that lends to abnormal or excessive cell growth, right? And there's all sorts of different potentials. Some cancers are slow, some cancers are very rapid to grow. It truly depends, all right? And we also can talk about cell growth and these tumors as benign or malignant. Benign tumors are non-cancerous. This is still excessive or abnormal cell division, but those cells don't cause issues. They don't impact the neighboring cells, right? The malignant, the cancerous situation is different, and they do impact neighboring cells and ultimately cause their demise as they continue to grow, right? There are other potentials, and I don't recall if we've got more of that. No, there's such a small amount here. We should have multiple slides with cancer, but we don't, right? There are other potentials with that when we think about these processes, right? So the cancers so malignant stuff can be growing in one location, and then it also has the potential to spread. We say metastasize. And when that happens, it basically breaks off from wherever it was initially and spreads throughout the body, potentially to very completely different areas or organs from where it had initially originated. Oftentimes, when cancerous cells have metastasized, that sets the stage for the prognosis to be pretty poor. It's much more difficult to treat when it's moved throughout the body, right? So there are a handful of things, right, that start to happen in this, although I don't have multiple slides to really show. One of the first things as we think about the cell cycle kind of going haywire, as this happens, and we think about tumor growth initially, normal cells have all sorts of proteins and mechanisms to recognize who they, where they are and who they are near and know what's happening, right? And normal cells, when they come in contact with other cells, they stop growing. They don't push into the other cell's area, they stop, right? So that maintains normal situation. When we get mutations like this, and we start going crazy with the cell cycle, one of the first things that happens is these tumor cells basically no longer care if they are touching something or not, and it allows for multiple replications, and they keep proliferating. If they impact other cells or organs, that's when we think of it really being cancerous, right? So contact inhibition is where you touch another cell and you stop growing, and cancerous cells or tumor cells lose contact inhibition. So they don't care about that. And really it's more the cancerous cells that have lost this contact inhibition, and it allows them to progress in, all right? I like to use an analogy of neighbors, and you've maybe heard that fences make good neighbors, right? Maybe your neighbor's all fine and good. That's great. Maybe your neighbor is not, right? And that fence might help to make things so you don't have to see that neighbor, right? So let's just imagine this. You've got your yard, they have their yard, you've got a fence in between, everything's all fine and good. This is a normal situation, non-cancerous, right? Now let's imagine that your neighbor is a cancer, all right? Your neighbor's a butthole and you've got issues here. You don't recognize oftentimes these issues until it's much, much later, all right? So let's imagine this neighbor has decided that you've got some prime real estate and he wants some of it. And so slowly, he starts moving the fence into your yard. And at first it's an inch or two that's been moved over. You don't recognize any of it. You're not paying that close of attention, right? And then he gets a little more, right? A foot or 2 feet into your yard. Maybe you start to notice, maybe you don't really notice until all of a sudden you open up your back door and you hit your neighbor's fence, right? You're like, where did this come from? That's cancer, right? All of a sudden you have no yard left because the cancer took it all, right? The bad neighbor, right? That's kind of what happens. And if it can't be controlled and goes unchecked, that's really where we start thinking about it being cancerous as they impact these situations. Okay, so enough of that. I'm sure there are probably some more details that the text spouts off, but I think that's probably sufficient. Lastly, we want to look at, and I think this really is, that is the last bit of cell division. Everything that we've talked about has been eukaryotes. It's been like our own types of cells, right? We also want to think the littlest bit, one single slide, we want to think about bacteria for 1 second, all right? Prokaryotes, they replicate, they divide just like our cells. They need to replicate as well. They do it a little bit differently, all right? It's more simplified because, well, first off, the prokaryotes are more simple to begin with. They don't have all of the other organelle stuff that we see in eukaryotes. We don't need to worry about, you know, pulling it, well, they still need to pull in nutrients, but we don't need to worry about pulling in these nutrients to replicate all of the organelles, to get all of that other stuff set up so that we can separate into new cells in the first place. They pull in nutrients, they got an adequate supply, they replicate their DNA, and in turn, they split in half. That's basically what happens, all right? It depends truly on the type of bacteria, all right? But bacteria, for the most part, just a generalization, can replicate super rapidly, all right? A single bacterium can separate into 2 new cells in potentially 20 minutes, all right? And in the course of a day, as these cells go through this process of replication, you could have a little teeny flask full of some nutrients, some broth. You could put a bacterium into that flask, and technically you're going to be putting a lot more than 1 bacterium. But you could put 1 bacterium into that flask, and over the course of a single day, you would wind up with billions of cells as they replicate exponentially, 1 to 2, 2 to 4, 4 to 8. All of a sudden, that flask that was once clear or nearly see-through is very cloudy, very turbid with all of the growth that's in there, right? Our cells don't really replicate quickly like that, right? But we could back it up and think about when we first started, and that's a little bit different, right? When the egg and the sperm get fertilized, we start going through this cell cycle, right? And that little zygote, that little fertilized egg that is basically going to just replicate out of control, almost like bacteria do, until we start getting specialized cells and we start thinking about organ development, right? So that very first handful of cells are almost replicating as quickly as bacteria, right? And they synthesize their DNA, and then they move to mitosis. And the gaps, we kind of don't even worry about, all right? It's basically synthesize and separate over and over, and all of a sudden, we've got all of these stem cells that are replicating, and we've got a mass of cells. After a few weeks of this process, it slows considerably, right? Otherwise, in a situation like that where these cells replicate super quick, love could be in the air. 4 days later, you're hatching out a kid, right? That's probably not what you want to be having happen, right? I would think, all right? So different, all right? These guys, we don't need to go through any of those issues, all right? So our cell up here at the top, we have ample nutrients. We're pulling in all of this stuff, metabolizing, being a normal cell, and we've got enough nutrients that now we can replicate. So there's some details in here. We don't need to get super bogged down with it, but the chromosomes of bacteria are circular. Eukaryotes have linear chromosomes, so the way they replicate is a little bit different, and we will see that coming up, all right? So, like a rubber band, we can go in both directions around this rubber band. We start at an origin of replication, which is where this initiates copying the DNA, and it moves in both directions, creating two bands, right? There's a handful of proteins that help to facilitate the cell division. And it's kind of like, it's not the same, but it's kind of like the proteins that are involved in cytokinesis, where we pinch the eukaryotic cell in half, right? These FTZ proteins come together. They help to get the chromosomes, and there's a lot of stuff happening here, but the bacterial chromosome, we don't have all of these microtubules that help to pull and do whatever. The chromosomes get attached to the plasma membrane, and as the cell starts to elongate, as we're preparing to split, it helps to pull the separate chromosomes to kind of opposite sides. The FTZ proteins help to kind of facilitate the chromosomes getting up there, but they also are what really does this pinching, like the cytokinesis, and that's what we're looking at here. So the bacteria are they're not plant cells, but it's kind of like that where we see plasma membrane plus cell wall material. There's other substances that are getting replicated in here and helping to produce this. It's not Golgi apparatus dumping cellulose at the midline, but these other substances have been generated in this process as well. And as the FTZ ring starts to constrict it, it basically pinches and pulls into two new cells. So we started off as a single coccus, spherical bacterium. It elongates and almost looks sort of rod-like, but it's really just preparing to divide, and then it pinches and separates. It looks pretty similar to mitosis, as we see one cell into two new cells, right? It is not all of the same steps and pulling and separating like that, but it is a similar idea. And ultimately setting the stage for cell division in eukaryotes, as we think about it bumping up from these ancestral forms to the eukaryotes, as we think about more detailed, definitely more business to get that stuff going on. All right. So as we're back, separate chromosomes, these cells are copies. They are basically clones of one another, genetically identical. All right. And that is similar to our mitosis. All right. These cells replicate and they're exactly the same. Right. Bacteria, these cells replicate, they're exactly the same. Right. And in our own body cells, we want that to be happening. And in bacteria, technically, that's what they want to be having to happen as well. All right. That sets the stage to transition into our next idea. And we want to start thinking about differences between stuff like bacteria that we will say are asexual. And as they replicate one cell into two, genetically identical, And compare that to sexual organisms that have egg or sperm. And in turn, these things vary, right? They are genetically distinct, and that creates variation. When we think about that from a big picture, right, variation occurs in sexually reproducing organisms pretty easily. And variation in prokaryotes, asexual organisms, is pretty difficult, all right? And when we think about environment, that's maybe a big factor, right? Which is better? Well, I think both have advantages and maybe some disadvantages. It depends on how you look at it. Both must be pretty successful because both processes are still happening today, all right? And it just depends on what you are. If you're a little teeny bacterium replicating super quick and making exact copies of yourself is probably a great way to go about it, right? As long as the environment stays very similar, right? With that said, this is not just a finite thing. Bacteria do have the potentials through mutation to have some variation, and through picking up different genes, there are some ways that they can share stuff with one another. It's not sexual recombination, but they can share genes, right? Antibiotic resistance is a pretty good example of some of that stuff, all right? So it's not like they are just static and never able to change, but it is different, all right? Are there any questions about that stuff? Okay, silence. Silence means no. Okay, I need to close that down. Maybe. Bear with me, guys. I have too much stuff happening. Nope. Okay, now I think we're back in action. Maybe we're. Now I'm back in action. Okay. So this next bit, as I was saying, we want to think about some comparisons between sexual and asexual organisms. Initially, we will think mostly about sexual organisms as we move through here. It's kind of, the bacteria at the tail end of the last chapter. That's kind of how it will happen. So there are all sorts of variations. And as we started this class, we thought about, well, what is life? And oftentimes thinking maybe some sort of big mammal, maybe a hippo, maybe not. Truly kind of just depends on what you might be thinking about. Every one of these things that we're looking at up here are multicellular organisms that are sexual. The way that they reproduce might be a little bit different, right? But all ultimately the same. And when it comes to plants, there's some pretty crazy variation with what we might see. And it depends on the type of plant that we're looking at. It could be a type of moss or, what do we have there? Some Joshua trees, I think is what these guys are right here. So and a bunch of other desert stuff. I'm sure there's some sagebrush up in there. We're pretty familiar with that here. So dependent upon the type of cell, there is all sorts of potentials with these guys, and often we think of possibly trillions of cells that we might see that make us, all right? So with, again, the sexual reproduction, there is a lot of possibilities, right? And this variation is super important, all right? So the last chapter, all of this should start making sense, all right? Mitosis, the same. And meiosis, the sexual generation of stuff, different, right? First guy same, second one completely different. And that is important when we think about variation, when we think about the potential to adapt to your environment, right? And deep down, that's evolution, right? You as a person aren't evolving, right? But as a population, we can see adaptations, right? Becoming better suited, we would hope, to our given conditions, right? So we want to think about this variation and the potential advantages that it might hold, all right? I've already kind of given us some hints about that. And then we will talk about some differences, life cycle strategies that we can see in multicellular organisms. There are also some similarities for sure as we progress along. So it really depends on the types of organisms that we think about as far as how they reproduce. And we can further divide some of this, maybe a little more thought about toward the tail end as we think kind of ecology, how populations are behaving in the first place. There are differences, right? And we're not really going to focus on that right now, but we can kind of think a little bit about it as we progress along. So we got the three different types broken down up here. A little bit difficult to see some of the stuff, but we can work through that. And you know, there's a few issues with this. The PowerPoints, some of these images are really kind of blurry or out of focus, but in the text, they are just fine. And I have tried with a handful of these to switch that stuff up and take it from the text and put it into the PowerPoint. But a few of them, that's not what has happened. So the book definitely is more clear if you are having some issues seeing some of this stuff. So the first one is us. All right, and other animals, but sexually reproducing adults will produce haploid gametes, egg or sperm cells, all right, and they produce these gametes from diploid germ cells, right? So the cells that are going to initiate this are like our own body cells with 2 copies of the chromosomes. But when we generate the egg or the sperm cells respectively, these things need to be haploid. They need to have half as much DNA, right? And when we talk, you know, the mitosis from the previous chapter, these cells replicate and separate those chromosomes so that each cell is genetically identical. They both have, for us, 46 copies. 23 pairs of these chromosomes, right? But in the egg or sperm, we want to take that 46 and chop it in half, right? So there are only 23 total chromosomes in the egg or the sperm. And upon fertilization, it brings us back to 46, right? If we did not do that, every time reproduction happened, the offspring would have twice as much DNA, right? And maybe one round of that could be okay, all right? Probably not, but maybe, all right? As soon as you get a handful of those rounds of replication, you got way too many instructions, all right? That just doesn't typically function, all right? There are some exceptions with that, and plants, they can do some kind of wonky stuff, right? There are numerous types of plants that have multiple copies of chromosomes, not just diploid, right? And in some of those circumstances, that could be a speciation event where they are all of a sudden different than what generated them, right? That's pretty impressive, right? You don't see that in animals, right? Diploid to haploid, and then haploid to diploid. That's how that occurs. So as we start with these people, right here, this is meiosis. That's what that is telling us. So it is the separation of these chromosomes within these germ cells to generate the haploid gametes, right? So as we go through this process, and we will see it shortly, it's going to look pretty similar to mitosis, right? There are a few things that happen in meiosis that don't happen in mitosis, right? And we ultimately, in the process of meiosis, kind of do this 2 times, right? That's really sort of the idea, right? To generate haploid cells, right? So through the process of meiosis, we generate these haploid gametes. They fuse egg and sperm to generate the zygote. That starts going through mitosis, as I described a second ago, really very rapidly initially. And then it slows down and starts to differentiate into all of the different types of organ systems, right? That brings us up to us as adults and the production of the gametes, the egg and sperm cells. This happens as puberty starts and egg and sperm cells start being generated, right? So that's the idea with this first bit, right? Diploid organisms producing haploid sex cells that generate diploid organisms, right? The organism itself is always diploid, right? We move down to some others and see some definite differences with this, right? And there are exceptions to some of this as well as we move through. Not everything's doing exactly like the fungi. In this case, not exactly like it, but most of them fit this category. So in this case, this fungus, Rhizopus nigricans, it's a black bread mold, something that most of us have probably seen at some point, moldy bread. So Rhizopus has haploid dominant stages and is diploid for just a brief period. We're different, right? So that's the idea. And here we've got multiple things happening here. This down below, that's the bulk of it, is the haploid stuff, right? When it is a single copy of that. And then up above is when it goes diploid. And this, like I said, there's some sort of oddities with this, all right? It is not the same idea as male and female and the fusion of these cells. But we do say with fungi, there are opposite mating types, and we say positive and negative, right? Like female and male, but it's not exactly the same, right? And when this stuff happens and they fuse, it's a double nuclei. It's called a dikaryon. And so technically it's diploid, but it's different, right? It's a little bit different. And upon this fusion, then we separate and we're haploid spores, right? That are being generated. So a little bit different, right? So they have the fungi, and there are variations with the fungi as well, but oftentimes we think of molds, lots of thread-like, hair-like projections. We call that hyphae. The hyphae of one type comes in contact with the hyphae of another type, That's where the fusion happens. Love is in the air with these little bread molds. And then they produce the spores. That's the kind of fuzzy, dark bits that you see. The black on the bread mold in this case are the spores. And within those spores are the haploid cells that will go back down to being the fuzz on the bread, right? Or whatever it's growing on, right? The bulk of the time, it is haploid, fuzz-like, right? Plants, there are all sorts of differences and huge variation when we think about plants, right? That's why we've got some here. There are some that alternate generations, which I think is pretty stinking cool. Mosses will do this. We bump it up here to ferns. That's a step up from the mosses. They both alternate generations, right? Both of which, dependent upon the generation, morphologically look very different, right? The one type of moss looks nothing like the other type of moss, and the same idea with the fern, which we've got the image of. So most of us probably have seen some ferns, and this is what we're seeing, the big leaves, oftentimes really pretty massive. On the underside of them, there's a bunch of little spores. The other generation, another point in time, it's a completely different looking plant, right? And the leaf is very broad. It's nothing like the fern leaf that we might imagine. And it would be the same if we were looking at some moss doing this, right? So at one point, the plant is completely different. It generates cells that are released, that grow up as the new generation of the plant, which produces spores, which are released, that grow up as this other generation. They are two completely different plants, but it's the same plant, all right? What the heck? Kind of crazy, right? And so they just generate different life cycle phases with that. And one point, the top one up here, the normal looking fern, if you will, it's this plant that is diploid. And it goes through meiosis and generates haploid cells. Those haploid cells come together, typically like moss and ferns both, need to be close to some moisture, close to a stream bank or something like that, a little puddle of water. And these spores can come together, they unite, and we form the next cell. So this one, generating the haploid stage and then to the diploid, right? Haploid, diploid. With that, as we're talking plants and some of the variation with this stuff. So mosses and ferns, we alternate generations. Then we bump it up and start thinking about seeds, right? Pollination. And a plant like a pine tree growing outside, it's a diploid plant, right? It's like us, diploid all of the time. And it produces the reproductive structures, the pine cones, and the pollen that we see, right? That fertilizes these cones. The cones are haploid, and they generate once they're fertilized, will generate a diploid tree if it were to grow, right? I grew up in Idaho. I've seen plenty of pine trees. I have noticed a handful of pine trees right around here. Walking down the sidewalk between the sports medicine and Holt Arena, there are a handful of pine trees, and I've seen numerous pine cones on these suckers. And I was pretty stinking sure that I knew what I was looking at when I saw these pine cones. I'm not a botanist, all right? But I saw these pine cones, and there are some pine cones that as I first looked at it, I'm like, oh, well, these are baby pine cones. They must be. And these are more of a mature pine cone. They look different, right? They have different looking appearances. This one pine cone's just not mature yet, right? That's what I thought. It wasn't until I started teaching this class that I learned that's not the case at all. The pine cones that look different, well, one of them's male and the other one's female. I'm like, what the hell? Who knew that? Botanists knew that, not me, right? So they are different, right? And in turn, Pine cones that are higher up on the tree, those are the male pine cones. They do look different. And in the spring, they release their pollen. They use the air to basically move that pollen around. You sometimes see it blown all over your car, right? The pollen blows off from the top pine cones and hopefully settles down on lower pine cones, usually pine cones maybe on a different tree next door, right? And it blows over to them. That's male higher, females lower, and they use gravity and the wind to help with the fertilization. What? Crazy, all right? The other ones, the female ones, tend to be a little sticky, and they are sticky, so as that pollen is blowing, it sticks to it, and there you go, all right? Oh my, who knew? Yes? That's a good question. I do not know the answer for that for sure. I feel like the female cone is going to be bigger than the male cone, but I could be wrong. Google it up. I'm pretty sure that the female cone is bigger. So that's pretty crazy, all right? And that first bit with stuff like the pine trees, the conifers, they just use the air, like I said. There's another type that uses pollinators, and we think about flowers instead of, you know, cones and things like that. The flowers rely on pollination typically from a pollinator. There are some potential exceptions to that. And they do it a little bit differently. We're not relying on the wind, and many of them want cross-pollination, some don't care, and it can be the same plant that pollination, or the same flower the pollination happens on. But they ensure that they have been pollinated before they ever produce any fruits, right? A pine tree makes all of these pine cones, doesn't matter, right? It makes male ones, it makes female ones, it hopes that the female ones get pollinated. Most of them probably do, all right? But some of them don't, all right? An apple tree doesn't give you any apples unless that flower has been pollinated. It doesn't waste its time pumping out a fruit if that fruit hasn't ultimately, if the flower hasn't been pollinated. Why waste your time to make the fruit? The seeds won't be viable, all right? So kind of different, and energetically, it's a big benefit for those flowering plants to ensure we don't waste our time unless we've actually been pollinated, all right? So a lot of variation with them, and mechanisms over evolutionary time that have led to their success. There are all sorts of different ones, and you know, equal levels of success based off of their specific habitats. The flowering plants, what we call angiosperms, have become the dominant plant that we see. They are very, very successful, right? Even here in Idaho. So meiosis, the generation of these sex cells, is what we want to think about as we progress through, all right? We're going to look at the chromosomes, how they interact. There are some, as I was describing, some things that are similar, and there is some stuff that is definitely different, and it's important because it's one of the instances that lends to some of this variation, right? So we'll go through the steps, see how some of that happens. We'll look at some differences between the two types, and then we'll talk some more about variation. So here we've got the chromosomes, and we talked before about, you know, if we're thinking ourselves, 2 copies, right? One from your mom, one from your dad, right? So this could be chromosome number 1, we would say, in our body. The blue one from your dad, the red one from your mom. The chromosomes replicate in the synthesis phase of interphase. We replicate our chromosomes. We pair them up. They're linked together by that centromere. And when they have replicated, they look like little X's, and we call them sister chromatids, right? So these sister chromatids, this is chromosome number 1 from your dad. This is chromosome number 1 from your mom, both of which have been replicated. So we've got the 2 sister chromatids here, and together we call them a homologous pair, right? The 2 copies, right? The homologs, right? And this image up here is one of the first hints of differences that we see in meiosis that you don't see in mitosis, right? When we replicate the chromosomes in mitosis, we don't pair the homologs up. We don't bring them together. We just line all the chromosomes up in a line, right? So all 46 of these guys are all lined up, and then we separate sister chromatids, and now we've got 2 identical cells. That's mitosis, right? Meiosis isn't like that. We want to go from diploid to haploid. And in order to do that, we got to have a little bit of control of what's happening. So we replicate, and then we pair up these homologs. There are other proteins that kind of help to hold them together. So now you get this group of 2 sister chromatids linked together, all right? That's a homologous pair. And you see this happening in meiosis, doesn't happen in mitosis. They would just all, like I said, all be in a line, right? They're still in a line, right? But the homologs paired together. So instead of 46, there's 23 duplicated, right? As these guys are held together, this is random. It doesn't happen all the time, and it doesn't happen with every chromosome, all right? But it can happen, all right? And this is important. As these homologous pairs are brought together, the sister chromatids have the potential to exchange material. These are the same chromosomes, right? 1 and 1, right? Genetically, they could be the same genes on there, both, dominant genes, both recessive genes, or maybe it's one of each. One good copy, one bad copy, if you will. Not that recessive is always bad, all right? So a good one, a bad one, or 2 goods, 2 bads, whatever it might be, however you want to describe it, those genes can have some variation, and we can exchange that. Now you've got a copy with the blue guy that's got a bit of red, and maybe the red gene on here is an advantage, right? And now you got a copy over here, and maybe this one's not, right? Or the other way around. It truly depends, right? And we could also describe it as this chromosome from your dad, right? Dad with a little bit of mom, mom with a little bit of dad, all right? Gives you a little bit of variation, some potentials that weren't there in you in the first place, but they are there in your egg or your sperm cells. All right, as we get some of this potential crossing over. So it is random. Not every round of this generates it, and it's not every single chromosome that we see all of this happening in. It is the ones that are held in the middle. These outer ones don't get the opportunity to cross over, all right? So you wind up, and as we go through all of these steps of meiosis, you will wind up with this side being in one cell, this one in another, so on and so forth. 4 different eggs or 4 different sperm cells, and 2 of which having some variation, different than what we started with, all right? That's important, all right? Lots of you guys probably have some siblings, and you might see some resemblance, and maybe you see some variation, right? Now we want to start thinking about some of this stuff, right? Is that variation the result of some of this? Possibly, right? There's some other things that also can lend to that, but this definitely can contribute to some of it, right? And if it's a lot of the same, it really kind of just depends on how these things were being separated in the first place, right? So when this has occurred, this crossing over, this genetic exchange, we call these 2 recombinant chromosomes because they're a little bit different than what we started with, right? And that's definitely an important contributor to the variation that we can see with sexual recombination, right? Giving us some genetic variation. And as we've got this on here real quickly, we are labeling genes, different letter designations. And there I mentioned good copies and bad copies, not really 100% accurate. Dominant and recessive, we will talk more details about, all right? The dominant genes we express with an uppercase letter, and the recessive genes we express with a lowercase letter, all right? So if you are homozygous, you have two of the same copies. And if you're heterozygous, you've got two different copies, all right? So this one right here, well, with this, because this is really you, we've got a big copy and a little copy. You would be heterozygous for this A gene, right? In these circumstances, it's all heterozygous. This individual is heterozygous with each of these, right? But if they were two big A's, homozygous dominant, two little a's, homozygous recessive, and a big A, little a, heterozygous, right? Depends on your genetic assortment, right? And we will go through and describe more of this in a minute. I just quickly, as we see the letters up there, that's what we're talking capital and lowercase, right? Upper and lowercase, right? Is dominant and recessive, right? And as you see here, this blue one that was all dominant stuff now has a recessive C, and the opposite with the reds, right? And maybe either confers advantage or possible disadvantage, depending upon the circumstances with that. So that's what we're thinking a little bit with that, is crossing over, contributing to some variation. Another potential is the independent assortment, as these things are being laid down. So you got two chances for how this stuff happens. And like I said, with mitosis, they all are just in one big line, separate the sister chromatids, right? What we're going to do with meiosis is bring the homologous pairs up first and then separate them. The next round will separate the sister chromatids, right? So as we're bringing these guys, blues and reds, into the mix, the first idea is, how do they line up in metaphase? Up on the top one, and we've simplified this, only throwing a couple of chromosomes in. But in the top one, the blues are all on one side, the reds are on the other, and as we separate, we get cells that are blue or red, right? In the other, it's different, right? So blue, red, then red, blue. And as we get this, we've got variation down here, all the same, different, right? Independent assortment, if you've got 23 pairs, and these 23 pairs are lining up, it is super unlikely that every single time that this happens, they all line up exactly the same. You're going to have some that get flip-flopped around. That in turn generates variation with the respective blue and red, how they line up in these cells. You still wind up with 23 when it's all said and done, but it's a matter of dad or mom copies that are being separated out. And then upon fertilization, you get the full assortment of all of that, right? And who knows what specific letters, uppercase or lowercase, you've got going on. Sometimes we know, all right? And sometimes you don't know, all right? There are some ways to evaluate some of that. So here we can see in the last slide, it was just straight up separation, right? In this case, we've also had some crossing over that's occurred, right? So there's other looking things. And in this one, we've had crossing over on the top and the bottom, all right? A little bit of exchange on both ends, and that's okay, all right? Different variation, right? It's also, again, just in the middle, all right? So When this happens, and we are just seeing the first little bit, right? But with this, we've got the blues on one side, the reds on the other, and we're going to separate. This is meiosis 1, and this is meiosis 2. Like I said, it's kind of like going through mitosis twice. It's not exact, but it's pretty close, right? So the homologous pairs are linked up, and we're going to separate them initially. That's what happens in the first round of meiosis. And we give the phases like we talked about in mitosis, same names, but prophase 1, and then in the next round, prophase 2. All right, just to tell you, it's happening twice. All right. So in the first round of meiosis, what we separate are the homologous pairs. We still have the blue with its sister chromatids together, right? And the red with its sister chromatids, right? They're still together, right? The next round of meiosis, after we have gone through all of the phases of 1, the next round is basically the same idea as mitosis, separating out these sister chromatids into separate cells, right? We've already cut it in half because we went from 2 copies, reds and blues, to just 1 of each, right? The reds or the blues, either way, all right? Does that make sense so far? Any issues or concerns? Okay. So here, we can see a comparison of the two, all right? Meiosis up on the top, mitosis down below. Remember, genetically different or distinct and genetically identical down below, all right? So as this happens, we take our cell, this first, as we're moving right here where I'm pointing out with interphase, this is that diploid cell, right? The diploid cell goes through the first round of meiosis and is going to become haploid, right? So prophase, prometaphase, metaphase, as we independently assort along this equatorial plane, blues or reds, vice versa, crossing over potentially has occurred. And if crossing over happens, it happens in prophase 1, all right? That's the only time that the homologous pairs are linked up and held together like that, prophase 1, all right? Crossing over can happen then. Metaphase 1 independently assorts as we potentially randomly mix these guys up along that plane. Anaphase 1 separates out the homologous pairs. Telophase pinches it off, a little bit of cytokinesis, and we're left with 2 cells at this point, haploid, right? These guys go through meiosis 2, separating the sister chromatids. This is very similar to what we're looking at below with mitosis, right? They line up, they separate, telophase forms a membrane around the chromosomes, and cytokinesis initiates where we pinch them off. And now we've got 4 cells that are haploid and they're genetically distinct, right? And with mitosis, 2 cells diploid, genetically identical, all right? So, as I've got this up here, all right, I want to quickly mention this, and then I will be done. I will let you guys out of here. What we are seeing on the top, that's really what happens in men, all right? When we see this process happening, we go through this process and we would generate 4 sperm cells, right? This happens in women as well, but you don't go through this process and generate 4 eggs, all right? I think you will say thankfully, all right? Because if that were the case, it would be pretty common, not pretty common, it would just be the norm that when you had a kid, you would have quadruplets, not a kid, all right? And, you know, I oftentimes ask some of this stuff, does anybody in here want twins? And I'm asking right now, does anybody? You're saying no, you're saying hello, right? Most people say no. Sometimes a girl will pipe up and she'll say, yeah. And I'm like, oh, do you have any kids yet? Of course not, right? If you had a kid and I asked you if you wanted twins, that would be a firm no, right? Because once you've got one, you're like two at the same time. I don't think that's an issue, all right? Four at the same time, forget about it, right? That would be trouble, all right? For sure. That's not what happens, right? Twins can happen. So can quadruplets, but typically there's fertility stuff that kind of lends to those situations. Not always, all right? So for the ladies, what is happening is a little bit different, all right? And I don't recall if there's another slide that goes through it or not, doesn't matter, I'm gonna tell you right now. As we go through this first round of meiosis and we generate these two cells, all right? One of these cells is gonna be a little bit bigger. It's gonna have more stuff, and the other one, not so much, right? The bigger one carries on to meiosis 2, and the smaller one does not. And it typically gets reabsorbed by the body. It gets degraded, right? Broken down, taken out of commission, all right? So now we're only dealing with one line, all right, in meiosis 2. We go through this round, we separate, segregate into separate cells, and the same thing happens. One of these, a little bit bigger, the other a little bit smaller, and the smaller one taken out of commission, and what you're left with is one haploid cell that you ovulate every month, all right? Again, there are some potential differences with that, and two might be released simultaneously. If that happens, you have twins, right? Those twins would be fraternal twins, different, non-identicals, right? Is that right? Is that what they're called? That's right. Yeah, whatever, it doesn't matter. You'd have twins that are non-identicals, all right? So same idea, just a little bit different with what's happening there. But I do think that's kind of important to think about, right? Because you don't generate 4 eggs a month, all right? I, again, think you would say, thankfully, all right? With that, I will stop. Any questions or concerns? Okay, guys, have a great weekend.
Thank you. Have a good weekend.
You too.